So microbes are the Earth's firstborn. They appeared on the Earth billions of years before plants and animals. And across this vast expanse of time, they've evolved a level of biodiversity that really challenges human comprehension. While some are pathogens, the vast majority are beneficial. And in their multitudes, these beneficial microbes help to sustain our biosphere. And they have profound impacts on plants and animals. So every environment contains a distinctive set of microorganisms which comprises its microbiome. So I study the microbiome of soil and its impact on the world we live in. So soil microbiomes are tremendously complex. They have billions of cells, even in a gram of soil, billions of cells, thousands of different species, and more genetic information than the entire human genome. So this vast biochemical diversity means that microbes can perform many reactions which are unavailable to plants and animals. Reactions such as nitrogen fixation, cellulose degradation, the synthesis of vitamins, and even synthesis of our antibiotics. So because they carry out all of these reactions, they're able to carry out many of the processes that help to sustain our biosphere. But many of these organisms remain poorly characterized. For example, the soil respiration response is very important to the carbon cycle. Annually, soil respiration accounts for 7 times more CO2 than all anthropogenic sources combined. And yet the soil respiration response and potential for sequestration of carbon in the soil remain some of our biggest unknowns for how the carbon cycle will respond to future scenarios of climate change. So if you think that doesn't matter, consider the fate of Biosphere 2. So Biosphere 2 is the first effort to make a completely self-contained ecosystem that could support human life. And it's a model for how we might one day colonize another world. So 8 people sealed themselves in this facility in a test of how well we could engineer a functioning biosphere. So they brought in fertile soil to sustain their crops so they could eat, but they underestimated the power of the soil microbiome. So these soils consumed oxygen, and they caused the CO2 levels to soar. So the system was out of balance, and within 16 months, it could no longer support human life. So these people would have died if they couldn't open the doors and get access to outside air. So we now know that soil microbes, and microbes in general, are critically important to the carbon and nitrogen cycle. So in my lab, we're interested in how microorganisms regulate nutrient cycles and influence plant growth. So I used to think that disentangling these relationships would be very difficult because of the vast biochemical complexity of soils. But advances in DNA sequencing technology and genomic technologies over the past 10 years have dropped the cost of DNA sequencing exponentially. And this has really launched microbiome science into the 21st century. This has been a game changer for everything that we do. And so in my lab, we're using these advanced technologies to explore the metabolic activities of thousands of microbial species as they occur in the soil. So we're discovering new species, and we're charting their activities in terms of regulating nutrient cycles. For example, we have found that major components of the carbon cycle are mediated by organisms that were largely unknown and uncharacterized before the use of these tools. So this information, this metabolic information, and understanding the nature of microbe-microbe interactions and microbe-plant interactions is important because it's allowing us to understand the basis of soil fertility. It'll allow us to investigate the sustainability of our management practices, and it's going to allow us to have tools for helping to improve degraded soils. All of this is going to be important information in terms of responding to the threats we expect from climate change. What's more, soils contain a vast reservoir of genomic diversity. So this matters because most of our antibiotics and many of our bioactive compounds were first isolated from soil organisms. And soils remain our greatest resource in terms of natural product discovery. These new genomic tools are really the resource we have to make future discoveries in this arena. We also know that microbes are critically important in terms of agriculture. So many microbes will colonize plants, and plant microbiome composition has fundamental impacts on plant biology and plant evolution. And we now know that, for example, if we look at fossils of plants, even the earliest fossils of plants, we see microbes in their roots. They've always been partners to plants. For example, many microbes will form mutualisms with plants, and these mutualisms allow the plant to expand its biochemical diversity, so it can get access to nutrients that it might not otherwise be able to use. For example, rhizobia are bacteria that form root nodules in plants, and these root nodules enhance soil fertility by allowing the plant to access nitrogen from the air. So we have opportunities to manipulate plant microbiome composition, for example, by putting the right microbes in a seed coating industrially, or using amendments or treatments that enhance beneficial microbes in the soil. The challenges we face are these are still very complex systems, and we're still only beginning to understand the rules that govern these associations. So advances in technology have really opened the door to microbiome studies. I believe that future studies of plant and microbiomes enabled by these technologies are going to facilitate future advances in understanding how to manage agriculture, how to generate improved plant varieties, but also allow us to make better predictions about how our biosphere is going to respond to climate change. Thanks.